initially. So considering before presenting your data, before completing your actual analysis of your data, what kind of data have you collected? What is the question that you're thinking of testing with that data? So what is the message? The main type of data that you're presenting, the main feature to be discussed. What's the message, how this information will be used, and of course, who your intended audience are going to be. And so when we think about tables, charts, and diagrams, which we'll come on to and think about in a little bit, we are also then assisting the reader with the data interpretation. So we want to produce figures, tables, charts, and diagrams that are representative of the data. And often authors don't do this. Often journal articles, you'll know yourselves from your own reading, from your own research, often journal articles don't do this. They obscure the meaning of the data by not presenting it effectively, by not presenting it accurately. So we don't want to do that. We want to help you to be as clear and effective as possible with your data presentation. Big mistake that people often make in these kinds of analyses are confusing dependent versus independent variables, of course, and bear in mind that these are used differently in statistical analysis than they may be in mathematics itself. So we are thinking here about a dependent variable being the event studied, right? So that's expected to change whenever an independent variable is altered. And so our ultimate goal as a researcher in our analyses is, of course, to find and determine the relationships between these variables. So be very clear, ask your colleagues, reach out to companies like ours, like Bentham, if you need help and assistance with these kinds of analytical tasks. Because if you then submit, people do this all the time, they submit an article to a journal that contains incorrect analyses or inappropriate statistical approaches to test the kind of question that they're interested in, a journal will quickly reject your article. So this, as a method, something to be very well aware of as you start to put your work together and send it to those international journals. So let's talk a little bit about some of these measures, starting with the median, the mean, and the mode. These are, of course, ways to estimate the center of a set of observations, to convey that one-liner information about your measurements. We often talk, don't we, about averages. So what do these actually mean? And you can reuse this information, you can share this information with students, with colleagues, definitions of these different attributes of your data as we get started. And of course, in real situations, outliers are common. And it's often, especially in my field, in biology, the outliers that are actually interesting parts of the data set. An outlying observation is an observation that's numerically distant to the rest of the data, for example, unusually large or unusually small. So this, of course, could be a measurement error, or that the population has a heavy tail distribution, or this could be interesting. Often we find, in particular in biology, that the outliers are the actual academically interesting attributes of our data sets. So we look at them in terms of symmetry as a data distribution. What are we looking at? Symmetry and skewness. A symmetrical distribution will have the same number of values above and below the mean, represented, of course, by the peak of the curve. And the mean and the median in a symmetrical distribution are going to be equal, aren't they? So outliers, you'll see in your own data, outliers create those skewed distributions. And so we're interested as researchers in determining positive or negative skew and describing what that means in the context of our research question. So describing the data overall, the centrality of the data, and then looking at how the data as a distribution would necessarily look, skewness and spatial variation. Moving on and getting more complicated, you would then look at the dispersion and variability in your data set, depending upon the kind of question that you wish to address with your data, of course. And as we've discussed, it's the mean that represents that central tendency, but on its own, the mean or the median or the mode will not really give us any idea of how the data is distributed. What we need to have to do that is an indication of data variability, the third of those basic statistical tests that we look at when we start to initially interrogate our data sets. So look at the range, look at the variance, and of course, most commonly looking at the standard deviation to give your readers, give your audience some idea of what your data variability looks like. So centrality, skewness, and now variability as we assess, as we interrogate our data sets. And how is this presented in academic articles? You see these kinds of sample distributions all the time in academic papers. What do they mean? You are, of course, subsampling in these cases from a larger data sample. So be careful with your notation, be careful with the way that you present your data. This, again, is another way in which often academic articles get rejected. Papers get sent to journals, they get sent out for peer review. The reviewer notices some fundamental issues with the data sample itself. So in this case, you can see the clear definition of standard deviation and what's going on with the notational confusion. And different people, different subjects may actually even use different definitions for the standard deviation. Is it something that's coming from a parent distribution or not? And are we going to be able, with the data that we've actually collected, to give an unbiased estimate of that parent distribution standard deviation from a given data subsample? Are you actually going to be able to say anything with your data set about that overall parental distribution? I know in my field, again, it's very often not possible. Paleontology, you have a limited sample from a limited time, limited locality, limited place in time and space. I can only talk about the variation and the skewness and the variability of my particular sample. I can't use that sample to say anything really about any other parental distribution. So that's something also to keep in mind that will vary depending upon your particular subject area. Moving on then, we also look at data variability in terms of quartiles and percentiles. These are very common approaches that colleagues use to subdivide their data set to describe their data set. So the quartile distributions, here you can see that's done at 25%, 50%, and 75%. And then you can look at things like the semi-interquartile range, the quartile deviation, or simply percentiles, the subdivision of data distributions that we're, of course, most familiar with because we use them all the time in common everyday life, 10%, 20%, and so on and so forth. But these, of course, have very specific meanings when you come to start describing your data sample in your presentation. So here is an example of the most common way, or one of the most common ways that people tend to present distributional data in graphical form. It's called a box and whisker plot. You've probably seen these, you've probably used these, how you can show quickly and easily what is the mean, what's the mode, what's the median, what's the data range distribution, and what are the standard deviations around that. So in this case, we're looking at a graph that shows the growth of guinea pig teeth, but it doesn't matter what the data set is. We are looking at how you can easily and quickly make these kinds of data presentations. So that's one that's very commonly applied. One of the biggest mistakes that people make, of course, and I know I've done this in my own research, confused this kind of thing in my own work. We see it all the time on TV. We see it all the time in news reports at the moment about COVID-19, for example, the current pandemic. Correlation versus causation. If you have a correlation in one of your data sets, a predictive relationship that can be exploited in practice. So determining the extent to which values of two variables are proportional to one another, you do not necessarily have causation. Proportion is something that's linearly related. So a correlation can be high if it can be summarized by a straight line sloped upwards or downwards. And of course, in statistics, this line, this correlation line, is called the regression or the least squared line. But I mean, just keep in mind that you may very often have correlations in your data set, covariance correlations, Pearson's correlations, the two main ways that these are developed and expressed statistically speaking. As you can see here, these are dependencies, looking at sample covariance, sample correlation coefficients. These are measures of correlations within a particular data set. Do they necessarily imply causation? That's the question. Often people, just because they see a correlation in their data, go on to make the leap of faith that that is actually related to some kind of cause. And of course, in our academic work, in our research work, we do have to be so very careful about getting across the message of our research correctly, but not overselling, not over talking up our results too much, because it may very well be the case that there is not actually a causation. Here are one of the main ways that people visualize correlation in data sets. Again, it comes back to the issue of data variability, data spread, using a regression coefficient, which of course, again, is something that most people have heard of, many people have used in their own research. And you can see here, comparing r equals 0 with r equals 0.9, what the different distributions look like. And clearly there's more of a correlation as the r statistic increases, the main way that this is done. So correlation and covariance, when looked at in a two variable state, we are then thinking about concepts of covariance. Correlation can be easily extended to arbitrary numbers of variables. So we can do this. I can show you how to do this. We can develop software. There is a range of software packages that you can use, and we'll talk about those in a minute, in order to develop what are called covariance or error matrices that simply look at the correlation between as many variables in your data set as you would like to look at, depending on the question that you are interested in answering with your data set. And as we discussed before, key point from this section of the presentation, correlation and causation are not necessarily the same. Be very careful. It's our responsibility as researchers not to present to our readers a causative relationship when one may not necessarily exist. Correlation does not imply cause. Correlation is a measure of linear relationship only. Misleading influence of a third variable is not taken into account. Spurious correlations of a part with the whole are not taken into account. We may be looking at the combination of an unlikely population. We may be making inferences based on an unlike population. So be careful with how you interpret your data. You can see on the cartoon an example here. You know, there may very well be an apparent correlation between two variables in your analysis, but have you selected those two variables for analysis correctly? Is there really a causative relationship between the two? And as you know, as we see a lot, often in research, these or inappropriate causative relationships are presented in academic papers, and then they get picked up by the media, and an inaccurate, inappropriate message gets spread out of academic research into the wider population. Here are some ways in which data can be presented in terms of determining that R coefficient, looking at things like least squared regression. I'm not going to go into this area in any detail in this webinar, but let me tell you that if you are interested in additional training, in additional statistical analytical training, perhaps for your university, perhaps for your research group, we can certainly provide that if you get in touch with us at the end of this presentation.